Welcome to our channel. In today's video, we're diving into the fascinating world of nucleotides, the tiny molecules that power our cells, store our genetic information, and drive countless biological processes. Whether you're a student, a science enthusiast, or just curious about how life works at the molecular level, this video is for you. Stay tuned as we explore their structure, synthesis, functions, and importance in health and disease. First, we need to know what are molecular building blocks. Molecular building blocks are the basic chemical units that form the larger, complex structures of living organisms. These building blocks include small organic molecules that assemble into macromolecules, which perform vital structural, functional, and regulatory roles in cells. They are essential for the composition of cellular structures and the maintenance of biochemical processes. Key categories of molecular building blocks: 1. Nucleotides. 2. Amino acids. 3. Monosaccharides, simple sugars. 4. Fatty acids and lipids. Each of these building blocks serves as the foundation for macromolecules such as DNA, RNA, proteins, carbohydrates, and lipids. Here we will discuss nucleotides in detail. Nucleotides, building blocks of DNA and RNA. Nucleotides are the molecular subunits that make up DNA, deoxyribonucleic acid, and RNA, ribonucleic acid, the essential macromolecules responsible for storing and transmitting genetic information in all living organisms. In addition to their roles in genetics, nucleotides are involved in cellular energy transfer, signaling, and enzyme function. Their versatile chemistry makes them indispensable to life. This comprehensive explanation delves into their structure, functions, and significance in biological systems. Structure of nucleotides. A nucleotide is composed of three main components. 1. Nitrogenous base. The nitrogenous base is a heterocyclic molecule containing nitrogen atoms. It provides the nucleotide with its unique identity. There are two types of nitrogenous bases: purines, double ring structures, including adenine, A, and guanine, G. Pyrimidines, single ring structures, including cytosine, C, thymine, T, in DNA, and uracil, U, in RNA. 2. Pentose sugar. The sugar component is a 5-carbon sugar. It differs between DNA and RNA. Deoxyribose in DNA lacks one oxygen atom at the second carbon. Ribose in RNA has a hydroxyl group at the second carbon. 3. Phosphate groups. A nucleotide contains one or more phosphate groups attached to the fifth carbon of the pentose sugar. The number of phosphate groups determines its form. Monophosphate, example, AMP, adenosine monophosphate. Diphosphate, example, ADP, adenosine diphosphate. Triphosphate, example, ATP, adenosine triphosphate. The combination of these three components gives nucleotides unique structural and chemical properties that enable their diverse functions. Nucleotides should not be confused with nucleosides, which are also 5-carbon sugar with a nitrogenous base. Nucleosides do not have a phosphate group. When a nucleoside is bound to a phosphate group, it yields a nucleotide. Thus, a nucleotide is also referred to as nucleoside monophosphate, if with only one phosphate group, nucleoside. Diphosphate, with two phosphate groups, or nucleoside triphosphate, when with three phosphate groups. Formation and linkage of nucleotides. Nucleotides polymerize to form nucleic acids, DNA, and RNA, through phosphodiester bonds, which link the 5' phosphate group of one nucleotide to the third hydroxyl group of another nucleotide. This forms a sugar phosphate backbone, with the nitrogenous bases projecting outward. The sequence of bases along the nucleic acid chain encodes genetic information. DNA, double-stranded, with complementary base pairing between purines and pyrimidines, are pairs with T, G pairs with C, through hydrogen bonds. RNA, single-stranded, with a pairing with U, instead of T, and G pairing with C. Functions of nucleotides. 1. Genetic. Information storage and transfer. DNA. DNA is the repository of genetic information. Its sequence of bases provides instructions for the synthesis of proteins and the regulation of cellular activities. DNA replication ensures the accurate transmission of genetic material to daughter cells. During cell division, RNA serves as the intermediary in gene expression. Types of RNA include messenger RNA, mRNA, carries genetic information from DNA to ribosomes for protein synthesis. Transfer RNA, tRNA, brings amino acids to the ribosome during translation. Ribosomal RNA, rRNA, forms the structural and functional core of ribosomes. 2. Energy transfer. Nucleotides, like ATP, adenosine triphosphate, are primary energy carriers in cells. ATP contains high energy phosphate bonds that, when hydrolyzed, release energy to drive various biochemical processes such as muscle contraction, active transport across membranes, enzyme activation. Other nucleotide-based energy molecules include GTP, guanosine triphosphate, which is used in signal transduction and protein synthesis. Nucleotides also involve in cellular signaling. Nucleotides play critical roles in intracellular and extracellular signaling pathways. CAMP, cyclic adenosine monophosphate, acts as a second messenger in signaling pathways, transmitting signals from hormones like adrenaline. The cyclic adenosine 3 and 5 monophosphate, CAMP, signal pathway, is a G-protein-mediate cascade of biochemical reactions associated with various hormone specificities and neurotransmitters. CAMP is a cyclic, second messenger, nucleotide that interacts directly with a handful of main targets: protein kinase A, pKa, cyclic nucleotide gated ion channels, CNGCs, and associated exchange proteins, EPAC. These pieces are linked to a number of vital downstream signal pathways, including metabolism, calcium handling, gene expression, vascular ability, cell proliferation, and apoptosis. CGMP, cyclic guanosine monophosphate. Cyclic guanosine monophosphate is similar to cyclic AMP, with the exception that instead of adenine, CGMP has a guanine nucleobase. It is derived from GTP, guanosine triphosphate. There are two major pathways of its synthesis, one via a membrane-bound guanylyl cyclase bound to a natriuretic peptide receptor, and the other a soluble guanylyl cyclase which is activated by nitric oxide. Like cyclic AMP, CGMP is degraded by phosphodisterases. Some phosphodisterases only affect CGMP, EG, PDE5A, the target of sildenafil, whereas others, PDE2 and PDE3, can hydrolyze both CAMP and CGMP. It's involved in a number of processes, including cardiac remodeling, blood pressure, vascular smooth muscle cell relaxation and contraction, anti-atherosclerosis, inherited, retinal degeneration. 4. Enzyme cofactors. Nucleotides form the core of several cofactors, essential for enzymatic activity. NAD, nicotinamide adenine dinucleotide. NAD consists of 1. Nicotinamide, active redox site. 2. Adenosine monophosphate, AMP, structural component. 3. Ribose sugars, link the nicotinamide and adenine groups. NAD involves in energy metabolism, redox reactions, cellular repair, biosynthetic pathways. FAD, flavin adenine dinucleotide. FAD consists of 1. Riboflavin, flavin mononucleotide or FMN, the active site for redox reactions. 2. Adenosine monophosphate, AMP, provides structural stability. FAD involves in electron transport, chain, oxidation of fatty acids and amino acids. Enzyme cofactor. Coenzyme A, CoA is composed of 3 main parts. 1. Pantothenic acid, vitamin B5. 2. Adenosine diphosphate. 3. Cysteamine, containing the reactive thiol group. It is essential coenzyme involved in numerous biochemical reactions, primarily in the metabolism of carbohydrates, fats, and proteins. Biosynthesis of nucleotides. The synthesis of nucleotides occurs through 2 primary pathways. De novo synthesis of purines. Purine nucleotide synthesis begins with PRPP, which, through a series of reactions, generates the nucleotide IMP. The first reaction is catalyzed by ribose 5-phosphate pyrophosphokinase to generate PRPP from ribose 5-phosphate and the 2 high-energy phosphates of ATP. Ribose 5-phosphate serves as a platform for the construction of the purine ring structure. PRPP is not committed just to purine synthesis. It is also used for other processes, including pyrimidine and histidine biosynthesis. The second step, catalyzed by PRPP glutamylamidinotransferase, commits PRPP to purine synthesis and is tightly regulated. In this step, there is a loss of the PRPP pyrophosphate and the addition of an amino group from glutamine. Then the steps of building the purine ring start. Glycine is added to form glycinamide ribonucleotide, GAR. Formyl group from tetrahydrofolate is added to GAR right-pointing arrow FGAR. Glutamine donates an amine group to FGAR right-pointing arrow. FGAM. Cyclization. FGAM converts into 5-aminoimidazole ribonucleotide, AIR. Carbon dioxide is added to form carboxyaminoimidazole ribonucleotide, CAIR. Aspartate donates an amine group to CAIR. Right-pointing arrow SAICAR. Fumarate is released, forming aminoimidazole carboxamide ribonucleotide, AICAR. Formyl group from tetrahydrofolate is added to AICAR right-pointing arrow FAICAR. Cyclization completes the purine ring, forming first nucleotide IMP. So, there are 9 enzymatic reactions that result in the formation of IMP, which contains the nitrogen base hypoxanthine. The pathway first builds the 5-membered ring and then the 6-membered ring. The amino acids glutamine, aspartate, and glycine serve as nitrogen donors, as ammonium, NH4, is not a nitrogen donor in biosynthetic reactions. The synthesis of IMP requires 5 moles of ATP, 2 moles of glutamine and formate, and 1 mole of glycine, CO2, and aspartate. The 4' moieties are transported on tetrahydrofolate, THF, in the form of N10-4-myle THF. The rate-limiting steps in purine biosynthesis are the first 2 steps of the pathway. The first step, catalyzed by ribose 5-pyrophosphokinase, is a feedback-inhibited reaction by ADP and GDP. The second reaction is catalyzed by PRPP amidotransferase. This reaction is also feedback-inhibited allosterically by binding of AMP, GMP, and IMP. Conversely, the activity of PRPP amidotransferase is stimulated by PRPP. Now the IMP can be converted into either AMP or GMP through distinct reactions. The formation of AMP requires GTP and production of GMP requires ATP. This allows for control of AMP or GMP depending on the availability of GTP or ATP, respectively. The accumulation of GTP results in AMP synthesis from IMP at the cost of GMP production. Conversely, excess ATP favors GMP production at the expense of AMP synthesis. Furthermore, AMP or GMP accumulation inhibits adenylosuccinate synthetase or IMP dehydrogenase to prevent further production of AMP or GMP, respectively. Subsequently, AMP or GMP can be converted to ADP or GDP by adenylate kinase or guanylate kinase using ATP, respectively. Because free ATP is present in higher levels than any other nucleotide, it is usually used as the phosphate donor in these reactions. Feedback inhibition of de novo purine biosynthesis by nucleotide. Purine synthesis is regulated by feedback inhibition. The first 2 enzymes of the pathway, PRPP synthetase and PRPP amidotransferase, are inhibited by purine nucleotides. In addition, the reactions leading from IMP to AMP and GMP are feedback inhibited by the end products. Clinical relevance. 1. Gout. Excessive purine catabolism leads to uric acid accumulation and deposition in joints. Treated with allopurinol, an inhibitor of xanthine oxidase, enzyme in purine catabolism. 2. Immunodeficiency disorders. Adenosine deaminase, ADA, deficiency leads to SCID. Severe combined immunodeficiency. Purine nucleoside phosphorylase, PNP, deficiency. Causes: immune dysfunction. 3. Cancer and chemotherapy. Drugs like methotrexate inhibit dihydrofolate reductase, blocking purine synthesis. Purine salvage pathway. Nucleotides can also be synthesized from the purine bases and purine nucleosides in a series of steps referred to as salvage pathways. Phosphorybosylation converts the free purine bases adenine, guanine, and hypoxanthine to their corresponding nucleotides. 2 key transferase enzymes are involved in the salvage of purines are adenosine phosphorybosyl transferase, APRT, which converts adenine to AMP, and hypoxanthine guanine phosphorybosyl transferase, HGPRT, which converts hypoxanthine to IMP or guanine to GMP by using PRPP as a substrate. By using PRPP as a substrate, the salvage pathway decreases the levels of PRPP, and therefore diminishes the rate of de novo purine synthesis. The de novo purine synthesis and salvage pathways are tightly coupled based on the availability of PRPP. One important salvage pathway is the purine nucleotide cycle, which produces the TCA cycle intermediate fumarate as a Mechanism to boost the TCA cycle's ability to generate more NADH for the production of ATP. An increase in muscle activity creates a high metabolic demand for ATP. An increase in AMP levels is a signal that energy generation by the TCA cycle and electron transport chain is insufficient to keep up with metabolic demand. This cycle is very important in muscle cells because they lack most of the enzymes of the major anaplerotic reactions, those that replenish pathway intermediates. Thus, muscle replenishes TCA cycle intermediates through fumarate generated by the purine nucleotide cycle. Myoadenylate deaminase is the muscle-specific isoenzyme of AMP deaminase, and deficiencies in myoadenylate deaminase lead to post-exercise fatigue and cramping. Clinical significance. 1. Lesch-Nyhan syndrome, caused by a deficiency of HGPRT, leads to excess hypoxanthine and guanine. Breakdown into uric acid leads to hyperuricemia, gout-like symptoms. Neurological abnormalities, including self-mutilation behaviors, inherited as an X-linked recessive disorder. 2. Gout, accumulation of uric acid due to excessive purine degradation. Salvage pathway defects, e.g., partial HGPRT deficiency, can exacerbate uric acid levels. Treated with allopurinol, an inhibitor of xanthine oxidase. 3. Cancer and chemotherapy. Purine analogues like 6-mercaptopurine and azathioprine are activated via the salvage pathway to inhibit nucleotide synthesis and DNA replication in rapidly dividing cells. Purine nucleotide degradation. Humans cannot break down the purine ring. The catabolism of purine nucleotides results in uric acid, which has low solubility in water and is excreted as sodium urate crystals in the urine. Purine catabolism does not alter the basic purine structure. The primary pathway is initiated by conversion of AMP to IMP or adenosine to generate inosine, which subsequently makes hypoxanthine. GMP can be converted to guanosine and, subsequently, into guanine. Both guanine and hypoxanthine generate xanthine. The final step involves converting xanthine to uric acid by xanthine oxidase, a step that produces hydrogen peroxide. Pyrimidine synthesis, de novo pathway. The de novo synthesis of pyrimidines is the process by which cells create pyrimidine nucleotides, cytosine, thymine, and uracil, from simple precursor molecules. Unlike purines, the pyrimidine ring is synthesized first and then attached to a ribose phosphate backbone. This pathway occurs primarily in the cytoplasm, with some steps involving the mitochondria. The steps in pyrimidine synthesis are: first step is formation of carbamoyl phosphate. The enzyme involved is carbamoyl phosphate synthetase 2, CPS2. The reaction is glutamine carbon dioxide 2 ATP produce carbamoyl phosphate glutamate 2 ADP phosphate. RIT occur in cytoplasm. This reaction inhibited by UTP, end product inhibition, while activated by ATP and PRPP. Second step is formation of carbamoyl aspartate. Catalyzed by aspartate transcarbamylase, ATCase, enzyme. The reaction is carbamoyl phosphate aspartate yield carbamoyl aspartate phosphate. The third step is cyclization to dihydroorotate. Enzyme involved is dihydroorotase. Carbamoyl aspartate form dihydroorotate via intramolecular cyclization. Fourth step is oxidation to orotate. enzyme dihydroorotate dehydrogenase catalyze the reaction. Dihydroorotate FAD produce orotate FADH2. Its location is in mitochondrial inner membrane. One thing to remember is that this is the only step in pyrimidine synthesis that occurs in the mitochondria. The fifth step is formation of orotidine monophosphate, OMP. Enzyme involved is orotate phosphoribosyl transferase. Reaction is orotate PRPP, phosphoribosyl pyrophosphate. Produce orotidine monophosphate, OMP, pyrophosphate. Sixth step is decarboxylation to uridine monophosphate, UMP. Enzyme is orotidine 5-phosphate decarboxylase. The reaction is orotidine monophosphate P to juice UMPCO. Formation of other pyrimidine nucleotides. UMP is the parent compound in the synthesis of cytidine and deoxycytidine phosphates and thymidine nucleotides, which are deoxyribonucleotides. Synthesis of cytidine nucleotides. CTP is synthesized from UTP by transfer of the amide nitrogen of glutamine to C4 of the pyrimidine ring of UTP. This reaction requires ATP as an energy source. The deoxycytidine phosphates result from reduction of CDP2. DCDP by a mechanism analogous to that described for the purine nucleotides. Then DCDP is converted to DCTP by nucleoside diphosphate kinase. De novo synthesis of thymidine nucleotide, DTMP. RIT starts with the formation of dump, UMP to UDP to DUDP and form dump. Enzyme involved is ribonucleotide reductase converts ribonucleotides, UDP, to deoxyribonucleotides, DUDP, followed by dephosphorylation to dump. Then conversion of dump to DTMP occurs. Reaction catalyzed by thymidylate synthase. Dump N5, N10 methylene tetrahydrofolate combine to form DTMP. Dihydrofolate. This step requires folate as a methyl donor. Regeneration of tetrahydrofolate. Enzyme involved is dihydrofolate reductase, DHFR. Dihydrofolate NADPH. Produce tetrahydrofolate, THF. THF is reused for further cycles. Regulation. Thymidylate. Synthase is tightly regulated to ensure a balance of DTMP for DNA synthesis. Antimetabolites. 4. Example 5-fluoracil, methotrexate, inhibit this pathway, blocking DNA replication. Clinical relevance. 1. Orotic aciduria, caused by deficiency of either orotate phosphoribosyl transferase or orotidine 5-phosphate decarboxylase. Its symptoms are megaloblastic anemia, unresponsive to folate and B12, growth retardation, excretion of orotic acid in urine. Treatment, oral uridine. Supplementation. 2. Anticancer and antimicrobial drugs, methotrexate and 5-fluoracil. 5-FU. Inhibit thymidylate synthase or dihydrofolate reductase to block DTMP synthesis, impairing DNA replication. Leflunomide inhibits dihydroorotate dehydrogenase, blocking pyrimidine synthesis in rapidly dividing cells, e.g., in autoimmune diseases. Salvage pathway of pyrimidines. The salvage pathway of pyrimidines is an energy-efficient process by which free pyrimidine bases, uracil, cytosine, and thymine, and nucleosides, uridine, cytidine, and thymidine, are recycled into their respective nucleotides. This pathway reduces the need for de novo synthesis, conserving cellular resources. Key features of pyrimidine salvage pathway. 1. Recycling of bases and nucleosides. Free pyrimidine bases and nucleosides are generated from RNA and DNA turnover or absorbed from dietary sources. These are converted into nucleotides, which are reused for nucleic acid synthesis or metabolic processes. 2. Primary substrates. Free pyrimidine bases, uracil, thymine, and cytosine. Pyrimidine nucleosides, uridine, cytidine, and thymidine. 3. Energy. Efficiency. The salvage pathway requires less energy compared to de novo synthesis. Steps in the pyrimidine salvage pathway. 1. Conversion of free pyrimidine bases to nucleotides. Key enzyme. Pyrimidine phosphoribosyl transferase, UPRT. Catalyzes the reaction between pyrimidine bases and PRPP, phosphoribosyl pyrophosphate, to form nucleotides. Reactions. Uracil PRPP produce UMP. Uridine monophosphate, PPI. Thymine PRPP leads to TMP, thymidine monophosphate, PPI. 2. Phosphorylation of nucleosides to nucleotides. Uridine cytidine kinase, UCK. Converts uridine and cytidine into their monophosphate forms. Reactions. Uridine ATP form UMP-ADP. Cytidine ATP form CMP-ADP. Thymidine kinase, TK. Converts thymidine into TMP. Reaction. Thymidine ATP. Produce TMP-ADP. Clinical significance. Disorders associated with pyrimidine salvage. Deficiency. Thymidine kinase 2-Tarkas deficiency affects mitochondrial DNA synthesis. Leads to myopathy and severe mitochondrial disorders. Use in chemotherapy. 5-fluorouracil, 5-FU. 5-FU is converted into 5-fluorodeoxyuridine via the salvage pathway, which inhibits thymidylate synthase, blocking DNA synthesis in cancer cells. Role in drug metabolism. Salvage enzymes can activate pyrimidine analogues used as antiviral or anticancer drugs, for example zidovudine for HIV, sotorabine for leukemia. Catabolism of pyrimidine nucleotides. Unlike the purine ring, the pyrimidine ring can be completely degraded. Pyrimidine bases are water-soluble, and the pyrimidine ring can undergo degradation to metabolites that can feed into metabolic pathways, such as the TCA cycle. Thus, pyrimidine salvage pathways are not as significant as purine salvage pathways. Catabolism of the pyrimidine nucleotides leads ultimately to beta-alanine or beta-aminoisobutyrate production, as well as NH3 and CO2. UMP and CMP are degraded to uracil and cytosine, which generate beta-alanine, which is subsequently converted to malonyl-CoA and used for fatty acid synthesis. DTMP is degraded to thymine, which is converted to beta-aminoisobutyrate, and next converted to methyl malonyl-CoA, and then to succinyl-CoA used in the TCA cycle. Beta-aminoisobutyrate and beta-alanine can also be excreted. Experimental applications of nucleotides. 1. DNA sequencing. The order of nucleotides in DNA is determined using techniques like Sanger sequencing or next-generation sequencing, NGS. 2. PCR, polymerase chain reaction. Nucleotides serve as substrates for DNA polymerase in amplifying specific DNA sequences. 3. Genetic engineering. Synthetic nucleotides are used in CRISPR and other gene editing technologies. Thank you for joining us on this journey into the world of nucleotides. We hope this video helped you understand their incredible role in biology and life itself. If you found this content informative, don't forget to like, share, and subscribe to our channel for more insights into the fascinating world of science.